Okay, so I just wanted to say a few words to introduce Kirsten. Kirsten Poff, who got her bachelor's at Eckerd College in marine science, so that well prepared her to do some of the work she did here at University of Hawaii. And then she went on to Texas Tech and got her master's, plant soil science. Actually, that prepared her very well to do some of the work she did here in Hawaii as well. And then in 2016, she arrived here in Manoa and started out in the laboratory at Gordon Bennett and then ended up working in our laboratory. She's had great research experience from all these institutions as a grad research institute, research assistant, at both Texas Tech and Hawaii, and an avian research intern when she was an undergraduate, and of course, graduate research assistant here at the University of Hawaii. And she's won a couple of awards along the way. Those are listed below. And one of the impressive things about Kirsten is her commitment to research and excellence in research. So these are Kirsten's current publications that are out there in the literature. They number around 8 right now, not including her master's thesis. So she's really done a lot of interesting and actually quite diverse work. Just to walk through that, some of Kirsten's research adventures include studying the ecology of fringed orchids in the southwest United States. Several publications as well as her master's degree resulted from those studies. And then here in Hawaii, she was involved in both studies of phytobiomes as well as insect microbiomes. And this, of course, was her work that she conducted when she started at UH, working with Gordon Bennett, and did some excellent work with him. And it was quite unfortunate when he left University of Hawaii, but we were really happy that Kirsten decided to join our lab. And of course, it was a logical thing to do. Kirsten took an obvious and logical trajectory, going from orchids to phytobiomes and insect microbiomes to trying to study some very complex features of the ocean microbiome. There is a link there and a connection, and it actually does make sense, even though it might seem like a stretch. So what else have we learned about Kirsten over the past few years? She enjoys really complex field projects. I'm not sure what you've got going on here, Kirsten, but I might ask you about that in the question and answer section. Okay. She's got, this one's serious. Kirsten really is skilled. She has, in the laboratory, her work is impeccable. She's very careful, and in her analyses as well. She's good in bioinformatics and computational biology, and she's even helped out this picture over here in keeping some of the servers going, disk swapping and that kind of thing. So hardware and software at some level. Kirsten really has a lot of skills. She's also a great team player, although as Uri here has learned, she likes to ride shotguns. So you lost that argument, Uri, but that's okay, because Kirsten, above all, in addition to being a great scientist, is a fantastic friend and a colleague and a mentor. And it's been just a joy for everyone here to have been working with Kirsten. And we're all excited to hear what she has to say. This is the title of her thesis, Dynamics of Planktonic and Sinking Particle-Associated Microbes in the North Pacific Subtropical Gyre. And with that, I'm going to stop sharing, and it's all yours, Kirsten.
Great. Thank you so much, Ed, for that amazing introduction, and thanks everyone for coming to my talk. There's way more people here than I thought there would be, which is really great. I'm excited to share this work with you all. As Ed said, my defense talk, Dynamics of Planktonic and Sinking Particle-Associated Microbes in the North Pacific Subtropical Gyre. So the North Pacific Subtropical Gyre, or the NPSG, is considered one of Earth's largest biomes at over 20 million square kilometers. Traditionally, this oligotrophic ecosystem has been thought of as sort of a static desert with very little biodiversity and very little variability, especially the area of the NPSG around the Hawaiian Islands. But the reality is that it's actually quite diverse. It's just that the organisms that live there are very small because of the oligotrophic conditions that they're subjected to. And it also does exhibit some seasonal variability, but this area of the ocean is still drastically understudied, in part because it's just so large. However, because of its size, chemical reactions from both abiotic and biotic sources have deep impacts on the Earth's atmosphere. So in 1988, Station ALOHA, which is a hydrological station that represents a 20-kilometer radius of ocean north of Oahu, was established. And since then, the Hawaii Ocean Time Series has sampled this area monthly to monitor short and long-term changes in this area of the ocean. So one of the long-term changes occurring in the ocean is ocean acidification, which is a direct consequence of increasing levels of carbon dioxide in Earth's atmosphere. So as you probably know, one important thing that the ocean does for us is it has the ability to sequester a huge amount of carbon from our atmosphere. Specifically, the oceans absorb about 2.5 gigatons of carbon from the atmosphere annually. And a large part of carbon sequestration by the oceans involves particles that form in the euphotic zone and are exported to depth via the biological carbon pump. But while these sinking particles are considered important, not a lot is understood about their origins, their composition, or their variability. And so while there's less seasonality in the NPSG around Station ALOHA than in more temperate areas of the ocean, seasons do drive production, which I will speak a bit more on later. However, another production driving feature common in the NPSG are mesoscale eddies, which are sometimes referred to as the weather of the ocean because of how they're able to transport heat and water masses. But essentially, mesoscale eddies are vortical circulations of around 100 kilometers that have timescales on the order of around a month. And we track eddies using satellite images and measuring their SLA, or sea level anomaly, which is basically just the deviation from the mean sea surface height. Eddies are ubiquitous and are formed by currents and winds. And depending on the direction the water is turning, either clockwise or anti-clockwise, a depression or elevation of deep water density layers or isopycnals will occur. So sea surface conditions can be simplified into three basic states based on SLA. Cyclonic conditions, where you have an anti-clockwise rotation, a negative SLA, and an uplifting of the pycnocline and deep water nutrients. Anticyclonic conditions, where you have a clockwise rotation, a positive SLA, and isopycnals and deep water nutrients are forced downward into the ocean's interior. And then finally, non-eddy conditions, where there isn't a circular current and SLA deviation is very small. So this depression and elevation of isopycnals, as I mentioned, causes a sort of downward depression or uplifting of nutrients and changes the depth of the deep chlorophyll maximum. So it has quite large ecological consequences. Some studies have found diatom blooms and enrichment or depletion of various bacterial groups like Prochlorococcus. Some work on sinking particles within eddies showed an enrichment in nitrogen fixing organisms in an anticyclone. But generally, while we understand that eddies can impact planktonic and particle associated communities throughout the water column, a majority of work done concerning eddy related changes in production is based on modeling or the sampling of a single cyclone or anticyclone, as opposed to an Eddy Dipole, which is essentially where you have a cyclone and an anticyclone side by side, So you have the ability to compare the two by either sampling a transect through them or sampling the center of the cyclone and the anticyclone. So that leads me to my three main research questions, which I will get into the specifics of as we go. The first being, how does the community composition of sinking particulate matter change annually? And what are the microbial drivers of elevated carbon flux events? Secondly, how do eddy-related shifts in pycnocline depth impact bacterioplankton populations? And then finally, how do particle-associated prokaryotic communities change across an Eddy dipole, and how are they correlated with sea level anomaly? So specifically for this first question, I investigated the community composition of sinking particulate matter at 4,000 meters at Station ALOHA, and also the microbial drivers of the summer export pulse event. And I attempted to answer these questions in my first data chapter titled Microbial Dynamics of Elevated Carbon Flux in the Open Oceans Abyss. So some background for you. The summer export pulse is one particular type of carbon sequestration event measured at Station ALOHA, which is basically this large, rapid, and predictable summertime export of carbon, nitrogen, phosphorus, and biogenic silica. And it results in a 150% or more, actually oftentimes it's a threefold increase in carbon export to the abyssal zone. And the theory is that photoperiodism causes life history changes in phytoplankton that cause this increase in deep sea export. So this figure on the right is from Carl et al.'s 2012 paper first describing this event and showing this stark increase in carbon occurring during the summertime. And so preliminary data from the Carl et al. 2012 paper, as well as other papers, have described a possible role in the summer export pulse by diatoms and their associates. Evidence for this firstly includes that increase in biogenic silica that I mentioned previously. And then in addition, in the 2012 landmark paper, they describe an increase in NPSG genes according to qPCR during the summer export pulse. And so this may be caused by nitrogen fixing symbionts housed by rhizosalinid diatoms that form these large balloons in the NPSG during the summer. So in order to do this project, samples were collected over the course of 3 years, 2014, 15, and 16. Sinking particles were collected by these sequential sediment traps shown on the right that were moored at 4,000 meters and operated for about 8 months each year, with samples collected about once every 10 days. And again, the objectives for this project were to characterize the community of the sediment and to correlate organisms and their metabolisms to elevated carbon flux events, especially the summer export pulse event, but also this secondary ECF event that occurred during the spring 2015 sampling. And so to do this, metagenomes and metatranscriptomes were generated from DNA and RNA extracted from the collected sediment in order to look at taxonomy and metabolisms. And right away, what we found in the ribosomal rRNA derived from the metatranscriptomes is that the bacterial communities on sinking particles are dominated by ultramonadales, shown here in green. And we think these are deep sea residents that are likely colonizing particles at depth. Those ultramonadales organisms were mostly members of Moritella, Coelia, and Shewanella. And Archobacter was also highly abundant, shown here in red, making up a huge percentage of the total metatranscriptome reads. But then if you take a look at the samples collected during the summer export pulse, shown as samples with a gray background in the geochemical data generated by the Carl lab, or outlined in black in the metatranscriptome reads, those samples have higher diversity than other samples in the data set, and they also have a higher abundance of groups that may be bloom associated, such as Bacteroidetes and alpha proteobacteria. The eukaryotic community was fairly variable throughout sampling. Lots of Cercozoa and Oligohymenophora. And then there were these sort of sporadic pulses of Cnidaria, Mollusca, and other opisthocons. And so, as I mentioned, there were two different data sets used for this project. The metatranscriptome derived rRNA, so that's rRNA extracted from the metatranscriptomes, and also Metagenome Assembled Genomes, or MAGs. So before I go on, I just want to get everyone on the same page so that it's clear when I'm referring to amplicon sequencing or MAGs throughout this presentation. So both of these approaches start with DNA. In this current chapter, I'm discussing, actually, we utilized a third type of sequencing using RNA, which is how we ended up with the rRNA derived from the metatranscriptomes. But generally, when you perform amplicon sequencing, you choose a specific gene and you use PCR to amplify that specific gene. In my case, I used 16S rRNA, so that you end up with multiple copies of the same gene you can use to identify organisms. Amplicon sequencing will provide you with a really nice sort of snapshot of community composition. And then shotgun sequencing, on the other hand, is not gene specific. So you wind up with all of these non-specific fragments from all over the DNA. And I will refer to those fragments as metagenomic reads. But then you can take those fragments and assemble them into whole genomes, or metagenome assembled genomes, or MAGs, which provide you with a really detailed look at some of the organisms in the community. And so jumping back in, one of the things that was done with both types of analyses, with both data types, was to perform a weighted gene co-expression network analysis, or WGCNA, to investigate which specific groups of taxa were correlated with increases in carbon flux. And we found that with both types of data, the same groups of bacteria were found correlating with increases in carbon flux, including Bacteroidetes, gamma proteobacteria, cyanobacteria, and others. And then in the eukaryotic rRNAs, we found a high abundance of diatoms and chlorophytes. So after looking at organisms that correlated with increases in carbon flux, we also wanted to look at organisms correlating with different elevated carbon flux events. So we performed several analyses looking at samples collected during the summer export pulse, and also samples collected during that secondary ECF event that occurred in the early spring 2015. And we found specific MAGs were found to be correlated with different elevated carbon flux events. So 40 MAGs were found associating with the summer export pulse, including groups of cyanobacteria, alpha proteobacteria, and other groups common in surface waters. And then another 7 were found to associate with that early spring 2015 ECF event. We also found specific metabolisms correlating with the summer export pulse period that imply surface origins, including photosystem 1 and 2, anoxygenic photosynthesis, as well as nitrogen fixation. And so here on the left is a figure showing the abundance of the MAGs that encode those pathways that I just mentioned. And one of the main organisms here contributing to an increase in photosystem 1 and 2 and nitrogen fixation is shown in orange on the graphs, is Richelia, which is a nitrogen fixing symbiont of Rhizosolinea, the diatoms previously thought to be responsible for a large part of the summer export pulse. And this rRNA-based figure on the right is the result of several network analyses attempting to understand prokaryote, eukaryote co-occurrence patterns in the sinking particulate matter. And this resulting network is showing only taxa which have the strongest interdomain associations and the strongest associations with elevated carbon. And right in the middle of the network, you can see rhizosolenids. They're central to the network along with groups of cyanobacteria, as well as Bacteroidetes, and then many of the same types of groups that we picked up with our WGCNA. And so this is the last figure I want to show you. It's a diagram for the different periods that we examined, and it includes a concise description of the communities on sinking particulate matter during these times, and then conceptually a bit about the processes we think could be responsible for the patterns that we're seeing. And there's a lot of information here, but to summarize, during the summer export pulse, we found diatoms and nitrogen fixers associating along with alpha proteobacteria, gamma proteobacteria, and Bacteroidetes. And some of the processes we think are responsible for this assemblage include those diatom diazotroph blooms, in general algal blooms, and epiphytic bacterial growth that is causing a rapid particle flux to the abyss. In contrast, the spring elevated carbon flux event, we found increases in acantharia, fungi, and copepods, along with a slew of prokaryotic traits that are likely due to things like copepod grazing, fungal colonization, and particle degradation at depth, and others. And then finally, during periods where particulate carbon wasn't elevated, we saw traits like parasitic and bacterivorous protists, as well as our altermonidales members, those deep sea dwellers. So in summary, these results support previous work suggesting that rhizosolenid diatoms and their nitrogen fixing symbionts are large contributors to the summer export pulse. But more than that, the MAG analysis is showing enrichment of metabolic pathways, reflecting surface origins, and the metatranscriptome analysis provides a thorough description of the community during the summer export pulse, that early spring ECF event, as well as non-elevated carbon flux periods. And it's just amazing, honestly, that we found all of these surface originating groups and pathways in this highly degraded sediment collected at 4,000 meters. And that brings us to my second research objective, which was to investigate how eddy-related shifts in pycnocline depth impact the bacterioplankton community. And specifically for this chapter, I focused on Prochlorococcus populations because preliminary analysis showed a possible difference in the communities between the cyclonic and anticyclonic eddy samples with regards to Prochlorococcus. And so my second data chapter is titled Differential Distribution of Prochlorococcus ecotypes in eddies of opposite polarity and over time at Station ALOHA in the North Pacific Subtropical Gyre. And so Prochlorococcus is an ecologically significant genus for multiple reasons. First, it's super abundant. It's also the smallest known phototroph, giving it an obvious advantage in oligotrophic systems. And finally, it's a super diverse genus. Its core genome is made up of about 1,200 genes, but its pangenome is estimated to be around 80,000 genes. It's also divided into many genetically distinct ecotypes, the two main ecotypes being highlight and lowlight groups. And then there are many ecotypes among those that are differentiated from each other by differences in genome size, GC content, ITS and 16S rRNA identity, as well as adaptive differences. And so at Station ALOHA, we already know quite a bit about Prochlorococcus dynamics. Cell abundances peak in the winter. And the dominant ecotype is highlight 2, shown on the graph on the right in yellow. We also know that generally these ecotype abundances are fairly stable over time. And highlight groups are good at photoacclimating over short periods of time rather than changing their relative abundances. And so for this work, I leveraged data collected on 2 oceanographic cruises, the 2016 Dipole and the 2017 Mesoscope cruise. During these cruises, high resolution sampling was performed in 3 locations across Eddy Dipoles, in the center of the cyclones, the center of the anticyclones, and then also the front between the eddies was sampled. And then from these water samples, 16S rRNA amplicon sequences and metagenomes were generated with, again, the objectives to compare Prochlorococcus communities across 2 adjacent eddies of different polarities, and then also compare Prochlorococcus communities during eddy and non-eddy conditions. And the way I did that comparison between eddy and non-eddy conditions was to utilize another data set. This one, an amazing data set collected by the HOT team. It spanned over 5 years with high resolution sampling occurring monthly at Station ALOHA. And so this first figure I want to show is the relative abundance of Prochlorococcus specific 16S rRNA reads from both research cruises. On the left are results from the 2017 cruise, and on the right are results from the 2016 cruise. And so when I process amplicon sequences, I use a denoising algorithm called DADA2 and separate the sequences into ASVs, or amplicon sequence variants, as opposed to OTUs, or operational taxonomic units. And these ASVs can be separated from each other with as little as 1 base pair change. And so each color on this bar chart represents an amplicon sequence variant, or ASV, which is just very fine scale. And so at this fine scale, you can see this interesting pattern where in both the Mesoscope and Dipole crews, there is enrichment of a specific sequence shown in red occurring in the cyclones. And so I built a phylogeny with reference sequences of known ecotype affiliation. And according to the phylogeny, that enriched ASV belongs in the highlight 1 group. And so this is especially interesting because highlight 2 is generally much more abundant at Station ALOHA. And again, that pattern is considered to be pretty stable over time. However, historically, Prochlorococcus ecotypes were described using the internal transcribed spacer, or ITS region. So to validate our 16S ecotypes, I did a cloning experiment where I pooled the Mesoscope cyclone samples and cloned the 16S through ITS regions so that with Sanger sequencing, I could obtain full-length 16S ITS region sequences, and I could examine whether my 16S ecotypes had ITS linkages that match the historical ITS ecotypes, specifically to validate that highlight 1 enrichment from the cyclones. And so the large majority of Sanger sequences I ended up with after the cloning experiment were highlight sequences. There were a few in the low light clades, but most of these sequences in the low light clades shown in the phylogeny on the right are reference sequences. And interestingly, most of the sequences were highlight 1. About 60% were in the highlight 1 clade and 40% in the highlight 2. So this fits our hypothesis that those Mesoscope cyclone samples that these Sanger sequences were derived from are enriched in highlight 1. However, not every ITS sequence that had the highlight 1 ASV sequence linkage fell in the highlight 1 clade. Most did. About 77% of the ITS sequences, again with that specific highlight 1 ASV 16S linkage, were highlight 1, but about 23% had linkages that were highlight 2. And I know it's difficult to read these large trees, but I colored the ITS sequences that have the highlight 1 ASV as their 16S sequence in purple, just so you can get an idea of where on the tree they are. And then the reference sequences are just shown in gray. And so this story, it's just not that straightforward. We can't say that every highlight 1 ASV sequence matches with the highlight 1 ITS sequence, but many of them do. And so based on these results, I did some metagenomic read mapping with reference ITS sequences. And here are the results from that process. The cyclone is shown on the top right, and it was enriched with highlight 1 sequences compared to the anticyclone, where highlight 2 was much more common. Furthermore, the top 2 hits in the cyclone were from sequences derived from highlight 1 genomes. MED4, the genome that matches the ASV highlight 1 sequence exactly, and MIT 9515. Whereas in the anticyclone, those top hits belong to highlight 2 sequences. And so based on this pattern where highlight 1 is enriched in the cyclones, I wanted to answer the question of just how rare this enrichment is at Station ALOHA. And I performed a variety of analyses using the hot time series data. One of the things I did was more metagenomic read mapping with the ITS Prochlorococcus database sequences. And on the right of this slide is the hot time series read mapping results. And they look a lot more like the anticyclone results, with highlight 2 being the dominant ecotype throughout the 5-year time series. And so the only place we see this highlight 1 enrichment pattern is in the cyclone. And so we found this rare enrichment, and finally we would like to know, why is this pattern occurring in the cyclones? So one of the final analyses I performed with the metagenomes was I examined gene enrichment in the cyclone, and also examined genes that were strongly correlated with ecotype specific ITS read mapping results. And I ended up with this long list of genes, which I aggregated by KEGG ortholog. And then to examine enrichment, as I said, we calculated the difference over total and log ratios of the reads in the cyclones versus the anticyclones. And I also correlated these genes with our reference sequences, again, of known ecotype affiliation. And then when I applied cutoffs for these metrics, I ended up with a list of 38 genes that were enriched in the cyclones and also correlated with highlight 1 sequences. And among this list are several genes involved in nitrogen metabolism, including a couple nitrogen transporter genes and ammonium producing genes, as well as some genes that may point to general metabolic differences between the populations in the cyclone versus non-cyclone conditions. Specifically, genes that could be favorable in more productive areas of the ocean, such as those involved in vitamin uptake and compatible solute synthesis. And so to conclude, highlight 1 is enriched in the cyclones, and it's also enriched in temperate waters at higher latitudes. Highlight 2, on the other hand, is enriched during non-eddy conditions in the subtropics. So, I'm sorry, non-cyclonic conditions in the subtropics. So ecologically, I think highlight 1 is enriched because, first of all, of cooler temperatures in the cyclone. So the cyclone samples in our study were significantly cooler than the anticyclones. And this is based on previous studies showing a lower temperature growth range of highlight 1. And then based on the gene enrichment in the MESOCOPE cyclone, There's also evidence that suggests highlight one might fare better in areas of high production because temperate waters are also more productive, not just cooler. And so we found that Prochlorococcus varies consistently over time at Station ALOHA, as well as between Eddy features. There may be Eddy-related adaptive differences among specific sequence variants of Prochlorococcus. And specifically, highlight 1 appears to be enriched in cyclones, which may be due to temperature, but possibly also differences in production. And so this leads us to my final research objective. How do particle-associated prokaryotic communities change across an Eddy Dipole, and how are they correlated with sea level anomaly? And specifically for this chapter, I used sinking particle samples collected at 150 meters on the 2017 Mesoscope cruise. And so my final data chapter is titled Impacts of Sea Level Anomaly on the Bacterial Communities of Sinking Particles. And so, as I mentioned, the sediment for this project was collected by particle interceptor traps, or PITs, deployed at 150 meters along a transect from the cyclone center to the anticyclone center in the Mesoscope cruise. These traps were deployed for about 10 to 12 days, just following the circular currents of the cyclone and the anticyclone. And I just want to take a second to acknowledge the heroic sampling effort by Carl et al. And the Scope Ops team. They deployed these traps across this Eddy transect and then went and found and recovered them all after this Lagrangian sampling period. And for the most part, they stayed pretty well in their respective water masses. So thank you for that effort. But then from the sediment that was collected, 16S rRNA amplicon sequences were generated, as well as MAGs. And again, my objectives for this chapter were to examine how the prokaryotic communities change along the Eddy Dipole transect and how they're correlated with sea level anomaly, as well as particulate carbon and particulate nitrogen. And so this first figure I want to show is the relative abundance of the 16S rRNA sequences collapsed at the family level, along with some beautiful geochemical data generated by the Carl Lab. I also have the average SLA values throughout trap deployment on the x-axis, ranging from the cyclone center on the left to the anticyclone center on the right. And with the exception of one sample, the community is dominated by Pseudo-Ultramonadaceae, with the Pseudo-Ultramonas genus making up more than 70% of the reads. And for this amplicon data, I didn't find any strong correlations with particulate carbon or particulate nitrogen along this transect. But amazingly, there's really good agreement between the MAG coverages and the 16S rRNA data. Here on the right is the relative abundance of the MAG coverage. I recovered 18 high quality MAGs. Most of the coverage belongs to the 3 Pseudo-Ultramonas MAGs, with the exception of one sample in the anticyclone that is dominated by a strain of Ultramonas, just like in the amplicon results. And those samples are denoted by a red star. But what these MAG results are implying is that there are different strains of Pseudo-Ultramonas that are differentially abundant along the sampling transect. So next, in order to see if there were specific groups of ASVs that were correlated positively or negatively with sea level anomaly, particulate nitrogen, and particulate carbon, I did a WGCNA with the amplicon ASVs. And none of the modules were correlated with particulate carbon or particulate nitrogen, but one module was strongly correlated with the cyclone. So when I collapsed the ASVs by family, and looked at the community composition of the cyclone-correlated module versus the other 3 modules picked up by the WGCNA, most of the pseudo-ultramonas reads grouped into the cyclone-correlated module. So then I did some other correlation-based analyses to examine which MAGs and ASVs were correlated specifically with sea level anomaly. And according to the 3 analyses I used, Kendall, Tao, Pearson, and Spearman, a single MAG was correlated with negative SLA values, which I outlined on the heat map with a red bar. And a specific pseudo-ultramonas ASV, ASV2, was also highly correlated. And so when we look at the abundance of the correlated pseudo-ultramonas versus the other pseudo-ultramonas ASVs or MAGs, the ASV abundance is shown on the top graph and the MAGs are shown on the bottom. There does appear to be enrichment in the cyclone of the correlated pseudo-ultramonas sequences, shown on both plots in a light green color. And so, but since these 2 approaches are very different, amplicon methods versus MAGs, now I would like to know, are these the same organisms in both data sets correlating with the cyclone center? And so we used PROCA to get the functional annotations for the MAG genes and then pulled out 16S gene from that particular cyclone correlated MAG. And then with the MAG derived 16S sequence, I just did a simple muscle alignment to compare the sequences. And there was only 1 base pair difference between the ASV and the MAG 16S sequence. So I think that these are likely the same organism found, correlating with the cyclone in both data sets. And so previous studies reported a high abundance of pseudo-ultramonas in pit traps or in pits collected at 150 meters at Station ALOHA. So the enrichment on the genus level is not a new discovery. Also, the community across the dipole is dominated by pseudo-ultramonadaceae, so origin processes may be consistent, even though there is the enrichment of that specific strain in the cyclones. So why is this particular organism enriched in the cyclone? This pseudo-ultramonas may be associated with a higher organism. A lot of pseudo-ultramonas are. However, this genus is fairly diverse and associates with a whole range of organisms, including some prokaryotes as well as eukaryotes. Some are also part of the trichodesmium microbiome, and there was a high abundance of trichodesmium in the cyclone during the mesoscope cruise. This particular pseudo-ultramonas may be associating with a different organism that is enriched in the cyclone and not in the anticyclone. But it's just really nice to see this pattern, because if you look at the genus level as a whole, we saw that the communities across the dipole were dominated by this genus pseudo-ultramonas, but then on a finer scale, we saw this different particular organism or strain correlated with the cyclone. And so this really highlights the importance of these fine scale analyses where you look at MAGs and ASVs versus more traditional OTU methods. And it's amazing to be able to find this pattern with these 2 very different approaches, the ASVs as well as the MAGs. So this brings us to the wrap up. Again, for my first main objective, during the summer export pulse, we found diatoms and nitrogen fixers associating in addition to alpha proteobacteria, gamma proteobacteria, and Bacteroidetes. And we think that some of the processes responsible for this assemblage include, firstly, those diatom diazotroph blooms occurring in the NPSG during the summer, as well as just general algal blooms and epiphytic bacterial growth that just causes this rapid particle flux to the abyss. And then in the spring elevated carbon flux event, we found increases in Acantharia, fungi, copepods, along with prokaryotic assemblages that are likely due to things like copepod grazing, fungal colonization, and particle degradation at depth. And then finally, during periods where there was not an elevated carbon flux event, we saw traits like parasitic and bacterivorous protists, and as well as our ultramonodalis deep sea dwellers. And so in the future, there's a lot of directions that this work could go. First, with the data in hand, we could explore the role of the Archobacter genus in this work, simply because it made up such a huge proportion of reads. I'm sure it's ecologically important. And just generally either in situ or designed laboratory experiments investigating particle succession throughout the water column to gain a better understanding of the formation and degradation of these sinking particles. And then for my second objective, we discovered this enrichment of the highlight 1 Prochlorococcus ecotype in the cyclones. And the highlight 1 ecotype is also enriched in more temperate waters at higher latitudes. And based on our analyses, ecologically, I think highlight 1 is enriched firstly because of the cooler temperatures of the cyclone. And again, this is based on previous studies showing a lower temperature growth range of highlight 1 versus highlight 2. And then based on the gene enrichment in the MESOCOPE cyclone, there's evidence that suggests highlight 1 might fare better in areas of high production or higher production, because temperate waters, again, are more productive, not just cooler. But, you know, I think an important next step here will be combining the gene analysis with the transcripts to try to understand gene-based differences and differences in expression between these populations in the cyclones and anticyclones. And also, it may be possible to generate some strain-specific mags with the long-read nanopore sequencing. We did preliminarily attempt to do this without much luck, but with a more thorough sequencing approach and with newer nanopore kits that have lower error rates, we may be more successful in doing that. And then finally, for my third objective, across the dipole, there was not a lot of variation. The community was dominated by Pseudo-Ultramonas and Ultramonas, but while we saw the communities across the dipole were dominated by this genus, on a finer scale, we saw this different particular organism correlated with the cyclone. So this really highlights the importance of that fine scale analysis. However, there's some obvious next steps that could be taken here. First, investigating the ecological role of the cyclone-enriched Pseudo-Ultramonas by firstly examining gene pathways in that particular mag. And then in addition, since we think that these groups that are in high abundance in the pits may be associating with higher organisms, doing some interdomain analyses to try and infer correlations between prokaryotes and eukaryotes would be really nice. And so that brings my talk to a close. Of course, I have a ton of people to thank. First, I want to thank Ed, my advisor, for all of his support through this process. It's just been unbelievably great. And also to my incredible committee, thank you for your time and effort in improving my work. And to my lab members, you have all been so great. Thanks for your technical, emotional, and intellectual support and help over these past years. To Dave Carl, thank you for making working at Seymour even more special. To all the amazing members of the HOT program and Scope Ops team, thank you for all of your hard work and expertise. Thank you to my coworkers, Seymour Halle administration and facilities workers, as well as people from other parts of oceanography. You know, I am obviously really more of a marine biologist, of course, but because of all of your help and teachings, I was able to accomplish these projects that involved multiple facets of ocean sciences. Thank you to my family and friends. Thanks to the viewing party in my living room. I hope you're having a good time down there. Finally, I just want to acknowledge, of course, the generous financial support that funded this project. Scope and the Simons Foundation are amazing, and I feel so privileged to be a part of that group. So thank you so much. And with that, let's start a discussion.